Okay, hello. Hi, everyone. My name is Nazila Kamali. I'm an associate professor at Imperial College, Department of Chemistry. It's really great to be speaking with you today. The talk today is going to be on stimuli responsive covalent nanogels, which is something that my lab has been really kind of looking at over the last 5 years or so as a very versatile drug delivery platform. And so just going to show you a little bit of background about where we are. So the chemistry department at Imperial College actually operates across 2 sites in London. So the pictures on this slide show you the new campus. So this is the new White City campus, and our building is right at the end of the slide shown here. I'm just wondering whether you can see my cursor, by the way, or hello?
We can.
No? Okay, so
Yes, we can see it.
okay, all right. So thank you. And then on the next slide, I'll show you a little bit more about these 2 campuses that I'm referring to. And hopefully, technology has not been very good with me today. It's not changing the slide. So why is that? Hopefully, well, I don't have to keep doing this, but okay. So here you can see the 2 campuses. There is Hyde Park. I'm sure you're familiar with in London. We're right next to Hyde Park and right next to the Royal Albert Hall. And actually, all of our undergraduate teaching occurs in the South Kensington campus. And then all of our research now has moved to the new campus, which is the White City campus. And that's where we have 6 floors of all very multidisciplinary chemistry labs and groups. And happy to show you around if you're ever in town. Okay, so yep, so it seems like this is not working for me, so I'll just have to go using the manual way. So most of you are aware about some of these nanoparticles I'm showing on this slide. So the purpose of this slide is to show you all the clinically either approved or clinically being tested drug delivery platforms out there currently on a global scale. So at the top, we have the more, if you like, biologically compatible and organic-based nanoparticles, starting from liposomes and all the way from micelles and lipid nanoparticles, which we're all very familiar with by now, polymeric nanoparticles, polymer drug conjugates, and so forth. And at the bottom, we have the inorganic nanoparticle platforms, which are used for either imaging purposes, or in the case of the gold nanoparticles, they're the actual technology in the lateral flow tests for COVID-19 tests. And so something around at any given time, between 30 or 40 of these platforms have been translated. And these 30 or 40 approved platforms, rather, are probably anywhere in the region between 2,000 to 4,000 clinical trials that are taking place across the globe. Of course, a lot of this has been led by lipid nanoparticles. One of the themes about a lot of these particles is that they are self-assembled, and they're mostly self-assembled using pre-existing polymers or lipids. And so there's a lot of reliance on this sort of like self-assembly. Nanogels come into play because they are relatively new, although, I mean, the term nanogel it's been around for a while. It's been around more than 20 or 30 years, and initially coined by Kabanov, who was working on developing these sort of gels. And in our hands, nanogels have a slightly different meaning in the sense that we develop them to be fully covalent and fully cross-linked with a payload inside until they don't need to be. And we'll see a little bit more about that shortly. And I'll just have to click away, unfortunately. So nanogels, if you're familiar with them, they are quite a versatile platform in the sense that the clue is initially in the name. They are gels, so by nature they are swellable. They can be shrunk and they can be swollen depending on different sort of physical or chemical behavior, and they're quite amenable to the as being, you know, vaccines or diagnostic agents, and lots of different uses in drug delivery, sensing, and so forth. We believe they're quite versatile because you can really literally put anything in them, and you may have to just try a little bit of clever chemistry to incorporate, you know, different types of payloads. For example, if you wanted to put a small molecule drug inside of them, they might be slightly challenging because you'll have to probably conjugate the drug to the backbone in the nanogel. But if you wanted to incorporate biologics, they're almost ideal for biologics. They can retain water up to 40% of their volume, and they're quite biocompatible. They're very versatile in design in terms of their size and morphology, surface chemistry. You can make them stimuli responsive towards a triggered release, or you can just release the payload by diffusion. And we believe they're quite colloidally stable. Of course, this is dependent on how they're made. So whether they are cross-linked or whether they're just made based on ionic or electrostatic or hydrophobic interactions. But overall, they're quite having worked in polymers for a long time in terms of drug delivery, they seem to be the next kind of area where a lot of researchers are moving towards because of all these great properties that they offer. Okay, so next slide. Again, as I mentioned, they're quite versatile. This means that you can really kind of change the surface properties. You can modulate the size and add your peg and allow, everything that can happen with other types of nanoparticles can also happen with nanogels. And of course, they're based on different types of techniques to synthesize them. They're obviously biodegradable, except for the sort of backbone that may remain in situ, but the backbone can also be cleared if it's less than 10 nanometers in size. And of course, again, we can incorporate targeting ligands on the surface to allow these nanogels to be more specific towards targets in vivo. And there are different ways that they exist in terms of typical nanogels. They can have core shells, depending on the speed of polymerization or the different types of cross-linkers or monomers that you want to use. And as I mentioned, they can basically have different sort of coatings. And you can even make them hollow inside. So you just have aqueous environment. And again, all the sort of bells and whistles and tips and tricks of other types of platforms can be applied to nanogels as well. And one of my very talented PhD students recently wrote a very nice comprehensive review on these, so I refer you to that. So in a very just crude way, in terms of how they're prepared, so we could either synthesize the building blocks in terms of polymers and have electrostatic or hydrophobic interactions and self-assemble them, or we can have actual covalent physical cross-linking of two existing polymers together. There's another technique which is using cross-linkers and monomers, and this is what we really favor in my lab because we think it's quite flexible in design, and so you can in situ cross-link the nanogel together. Okay, so moving on to a little bit more of what my lab specializes in and we'd like to sort of get more and more into is the fact that we really do believe the first generation, if you like, of the drug delivery platforms that have even been translated to the clinic are non-targeted. So the drug is entrapped and then the drug is released usually through diffusion, especially if we're talking about polymeric drug delivery. And the second generation is the targeting nanoparticles, which have not done as greatly as we'd hoped so far. But that's the second generation. And the third generation is really the stimuli responsive, the sort of extra element of allowing the drug to be released in a more controlled manner. And so we look at a lot of chemistries that are amenable to exogenous stimuli, as shown on the left-hand side, and then also endogenous stimuli, which are in our hands a lot more interesting because if you consider different diseases, for example, inflammation has a lot of reactive oxygen species and free radicals around. Enzymes are upregulated in tumors and diseases, and pH is reduced inside the cell or within tumors as well. To an extent, but one molecule we're very much interested in using is glutathione. So glutathione is a housekeeping homeostasis sort of peptide inside of cells, and it's found in a much higher concentration, and it works really well in our hands. And so this is the sort of thinking in terms of developing this toolkit. So as you will see shortly, what's really nice about the flexibility in the design and synthesis of these nanogels is that you can basically pick your stimuli. You can use chemistry to create the heat sensitive nanogels, or use other types of linkers to create different sort of stimuli responsive nanogels, and they're quite versatile. So here, for example, we say we can develop pH responsive nanogels, and so a change in pH can either lead to swelling of the contents, or a change in pH can actually lead to the entire disintegration of the nanoparticle, and that's achieved, for example, by using chemistry that's responsive to acidic protons in solution. So all of these different types of linkages are acid labile. And so we're able to instill these in nanogels and allow them to sort of change their property, either disintegrate and release the payload, or they shrink and they release the payload through diffusion. So here we can see a snapshot of a very basic kind of method, if you like, that we use to make our nanogels. So we like to call this a nanopolymerization, if you like. So what we are able to do is create these nano droplets because we use micelles. So we're able to use micelles where within the core of the micelle, we can entrap most of our monomers and all the desirable features we need in our nano gel. We can then nanopolymerize it in situ, purify it, and then apply stimuli. And I'll talk a little bit more about what the crosslinker is, what the monomer is, what the charged, for example, monomer is, and the rest of the sort of like methodology. And in this way, actually, we're able to make these nanogels in a single step in water, so they're also sustainable, and it's a green one-pot synthesis, if you like. So this shows you a little bit more of the breakdown of these nanogels. Now, of course, the literature has many ways to make nanogels, and there are lots of leading groups in this field who've established a lot of different techniques, and some of these concepts are borrowed from other kind of like researchers where they've been using these sort of like cationic charged moieties a lot in terms of templating or creating sort of not cross-linked, but sort of polymerizable anchors, if you like. And so in our hands, the lab specializes in synthesizing cross-linkers. So we synthesize very precise cross-linkers that are quite short in nature. They are usually flanked by a very small repeat units of PEG, because we want the cross-linker to not have too much space to allow the payload to come out prematurely. So there is, for example, a very short peptide sequence we've explored for matrix metalloproteinase cleavage. And this peptide sequence is flanked by a very short PEG. And then if you've got the double bonds or methacrylates and acrylates, you're obviously polymerizing into a cross-link structure. So here, for example, we have cathepsin D. These are used in separate applications. I'm just kind of showing them to you in one go here. Cathepsin D cleavable monomers, and here like we use the disulfide, as I mentioned, which is really great in our hands because it's responsive to glutathione, and then pH responsive monomers as well. So when we choose the different type of monomer, we synthesize the cross-linker first, and then monomers in terms of acrylamide. Again, this is just if you've done a molecular biology lab experiment where you've created a gel in the microwave, same kind of thinking. So acrylamide is just a backbone sort of like space filling agent here. Charged monomers, we screen different types of charges. This is a quaternary amine based one, an ATC. It works quite well in our hands, but we've looked at lots of other types of charged monomers as well. And the payloads we've investigated have mostly been biologics, because the payloads in our hands, these are really kind of, where the next, again, sort of revolution in drug delivery is moving more and more towards biological payloads and biological therapies. We use the APS-TMED as a water-soluble initiator, but of course, other types of water-soluble initiators can be used. SDS here is the surfactant. So even though we are solvent-free, if we remain within the critical micelle concentration of the SDS, we are able to create these kind of like nano droplets, if you like, with the other constituents of these nanogels. And so we mix these together as shown. So this is just a typical example of a synthesis. I think if I maybe don't use this cursor, I might be able to change the slides, because a couple of these are going to come up. No, still not working. Okay. Okay, so now it might be okay. So here on the left, you can see the typical mixture, if you like, of the synthesis. So we're able to mix all of these components together and then polymerize them, creating a cross-linked and also a copolymerized backbone. And obviously, this is just the electron transfer shown here, how we initiate the free radical. This is a little bit more detail about how we are synthesizing these things. So the red dot can be your biological payload. So it could be a protein in this case. And then we electrostatically template this ATC around the protein. And then in situ, after about half an hour in water, we start to add the rest of the reagents and polymerize around this construct to give us usually about 100 nanometer nanoparticles after simple purification. Purification can be just simple amicon filtration or centrifugation and so forth. So, for example, to give you an sort of idea of how these things can work. So we took a nanogel with a cross-linker in the middle. The cross-linker had a MMP responsive peptide sequence in the middle, and then we were able to actually encapsulate a enzyme which is called paraoxidase 1, and paraoxidase 1 actually is the first step in the generation of atherosclerosis. So it can stop the oxidation of low-density lipoproteins into oxidized lipoproteins, which drives the plaque formation in atherosclerosis. And so we were able to show that we can actually drive this sort of chemistry and favorably change the macrophage biology and so forth. So it's quite a delicate platform for the delivery of these very super sensitive types of enzymes as well. And so okay, and on this slide, we're just showing you how, for example, if you have a monomer that is got a peptide sequence in the middle, the peptide sequence is built in a stepwise manner using solid phase peptide synthesis. So in this case, we just started with this commercially available alkene, and then we were able to use solid phase peptide synthesis to instill and build sequentially the peptide sequence we need, and then cleave it off resin to give us, like in this case, this cross-linker. Then with the cross-linker in hand, we do all the required testing. So we tested the matrix metalloprotein is responsive cleavage using MALDI, and mass spec techniques, and showed that we're indeed able to cleave the peptide sequence. We were then able to make nanogels. So 5.2 nanometers shown here is the protein alone. 19 nanometers is the empty nanogels, and 58 nanometers shown is the actual nanogel with the protein inside. And the morphology looks quite nice under TEM. And what's really nice and quite surprising, but actually is probably in my view, the unique selling point of these nanogels is the fact that they are really super stable. You can look at their size across time. 30 days, 40, 50 days, we've looked at these things, and the zeta potential, they're extremely stable because they are fully covalent. They're covalent systems. And I think that this is one of the key sort of applications and advantages of these systems, in that even the payload can remain stable within them until it needs to be released. So there's a very preliminary studies where we've looked at sort of the release. In this case, it's just bovine serum albumin of a protein. And so we can see the red line is a physiological concentration of matrix metalloproteinase enzyme, and it's led to much enhanced release of the payload. And here, these are the initial studies where the natural diffusion, the baseline is 20% out of the nanogel without the enzyme. But however, we have managed to now reduce this 20% to less than 5%. So we believe the 5% that's released from the nanogel before meeting an enzyme is because it's just surface bound or diffusion, just surface layers of the nanogel. And then we do the whole gauntlet of like cell toxicity experiments and uptake experiments. We look at the native behavior, like activity of the enzyme that's not seen a nanogel, and then one that's seen a nanogel and has been released. And you can see that even given the free radical conditions that we're under, we're not perturbing the activity of the enzyme as much. We can make them fluorescent and look at the encapsulation yield and so forth as well. So here you can see when we incubated them with macrophages. So these macrophages had eaten a lot of oxidized LDL, and then this is the treatment, the enzyme on its own, and this is the treatment with, I'm not sure you can see this cursor, but the top right, you can see the nanogel is behaving just as well as if we administered the enzyme on its own. And so the less red and purple we see, that means the less oxidation. And again, we show, the actual kind of measurements as well in terms of like being able to reduce foam cell formation, which is macrophages not eating as much oxidized LDL. And then we're able to show the concentration of the oxidized LDL is going down as well. And also we're inhibiting the activation of reactive oxygen species in this study. What was really nice is that we showed that these nanogels can actually also cross the blood-brain barrier. So we collaborated with Felicity Gavins in London, and she has some mice that have stroke induced within their brains. And so we were able to inject our nanoparticles, which in this case, after we put a peptide inside, because the peptide is anti-inflammatory, and the peptide made these nanogels even larger, maybe 250, sometimes 300 nanometers in size, but that didn't stop them from crossing the blood-brain barrier. And we were able to show that they are localizing, and we were able to show that we are favorably sort of driving the biology towards lowered inflammation, and the mice didn't really have any sort of bleeding or microbleeding over time. And we were able to show that our nanogels, which are shown here over time, could in green, can reduce the neurological score, which means how bad the ischemia is in the brain, and the rest of the biology as well. So we were able to show that the mice not only did they not die, but they were able to sort of sustain the therapy, and the peptide was released in situ. So that was very good news, but we wanted to move on to different systems, and then also to look at more, moving towards sort of nucleic acid delivery. So this is work done by a very talented student of mine, Rupali Davas, who is working on these nanogels for biologics and nucleic acid delivery. So she synthesized these different nanogels where we have a disulfide moiety in the middle, and control ones where we don't have a disulfide, and then even PEG that's elongated, sort of like, this is PEG about 45, which is much larger in design. And so she was able to use the same optimized in situ water-based polymerization technique to create these nanogels and then study them. So for each type of study, these are the sort of experiments that we perform. So we look at the sort of optimizing the percent monomer conversion as a function of, for example, the hydrodynamic size of these nanoparticles, and we'll image them by TEM. And we look at the encapsulation efficiency and the loading efficiency, and also the release of the nano, in this case, sorry, this is a model protein, a fluorescent protein over time. And we can see that this red one is the control that doesn't have this disulfide linkage that is amenable to control by glutathione. But the middle one, the green one, does. And then this one is with the longer PEG. The bottom one is the one that has the much larger PEG. And we can see that the much more defined and controllable release is this curve in the middle, the green one, where we can show that the release is indeed triggered in response to different concentrations of glutathione, which is the sort of stimuli, if you like. The one with a very long PEG is a little bit sort of not as predictable or controllable. That's understandable, because if you make the pores really large, then you're inherently causing a lot of kind of diffusive behavior, and the payload can come out, and the nanogel has more sort of like less controllable features. But this sort of middle feature, as you can see, is the middle parameters is what we chose to sort of move along with. So here we show again the stability on the left-hand side, which is size versus the polydispersity of these nanoparticles over a 7-day period. So both the top ones did quite well. The bottom one that had the much larger sort of peg chain was a bit higher in size and swelled a bit over time. On the top right here, we can see some uptake studies, just some simple uptake studies inside cells to show that the nanogels get taken up. The middle panel here, we can see a circular dichroism study where we look at, for example, the naked protein on its own, which has not seen a nanogel, and then the released, the reaction mixture condition, the released protein, and the protein stored after 30 days in the nanogels. And you can see that the CD data looks quite similar. It looks almost identical to the red line, which is the naked BSA. And then here we took our paraoxenase enzyme from the previous study. We also wanted to look at its encapsulation, its release, and again also looked at sort of what all the different conditions mean. And in our hands, we were able to show that we do see a little bit a little bit more loss of activity, but not that much more loss of activity. So it's still working in these conditions. And we were able to also like measure this and quantify this effect as well. So next, we looked at what can we do. We've proven we can put proteins inside, but what can we do more functional payloads? Can we look at sort of nucleic acids? And as many of you know, the lipid nanoparticle platform is huge. It's a billion dollar industry. Every pharma company wants to have an LNP. And the lipid nanoparticles, which are the basis for the Pfizer and Moderna vaccines, have done great. You know, they've been in 18, 19, 20 billion patients times administered into people, so they're quite safe. But we believe that, you know, there's still room for improvement. So because these are based on vaccine delivery, and they have a 50% cationic head group lipid within them that can cause toxicity. This minimizes repeat dosing. So you can only really inject these once every 6 months or once a year. And so again, we want to try and mitigate some of these problems as listed here that are currently ongoing, you know, with the lipid nanoparticles. So for example, issues around this repeat dosing, which makes them very good as vaccines, but it may not make them ideal as therapies, so in terms of regular use therapies. So we want to look at this sort of improving on endosomal escape, immune response, as there's a cold chain associated with these things. So can we just, you know, avoid the very cold temperatures needed? And there's stability issues in terms of lipid oxidation, lipid aggregation over time, very limited LNP chemical design, although now with the whole big kind of AI, ML-based approaches, we might be seeing different things. And the IP space being crowded, I mean, patents are going to start expiring. So there's a need for lots of different formulations and designs. So again, Rupali led this work. So as I mentioned, glutathione is this natural kind of like a tripeptide, which is found inside of cells in a much higher concentration compared to extracellularly, so 1,000-fold more found inside of cells. And what this means is that if we instill disulfide bonds within the cross-linkers, we should be able to easily cleave them. And there's a whole equilibria reaction that happens. And so we get, you know, free thiols at the end. And so Rupali synthesized some monomers as shown here. So this is our nanoparticle, our nano polymerization sort of technique. And so our A will always be acrylamide, excuse me, our B will be a positively charged moiety, and C is the cross-linker. So she was able to design and synthesize similar nanogels for this purpose. And she was then, so this is the exact composition, for example, of the solution, if you like, in water, where we again, in a single self-assembly step, are able to entrap mRNA. So in this case, we looked at GFP mRNA, which is a simple mRNA to use. And then she studied, for example, again, the ATC-MOL conversion over time as a function of zeta potential, looked at the encapsulation and loading efficiency, and then release, which is really nice. So we can see that RNA releases over time in response to physiological concentration of glutathione. and that's the red dotted line shown here, but whereas the baseline is no glutathione. So you can really hopefully appreciate that these covalently sort of cross-linked nanogels can protect the payload until the disulfide bonds are cleaved. So this was really nice studies, and then she carried on to sort of really look at what happens over time. So this is time 0 TEM of these nanogels, and then after 6 hours exposure to glutathione, 12 hours and 20 hours, and at 20 hours obviously there's a kind of like a big mess of aggregation and so forth happening. This is just a dynamic hydrodynamic sort of size measurement of the same sort of thing that we're seeing here. But on the other hand, when she looked at under TEM of the samples that have not seen glutathione, you can see that they're quite stable. So overall, the TEMs look pretty stable and really uniform throughout. So that's all the red dots shown here. Next, she looked at release. So, of course, there's 20 micromolar extracellular concentration of glutathione in the body usually. So she could show that there is some release, premature release of the RNA, but this could also be surface-bound RNA as well, because you have this ATC positively charged monomer there. But on the right-hand side is a different story. So now we are controlling the RNA and in a lot better manner, 82% released in response to the glutathione concentration, which is found inside of cells. So in a way, we are creating these to be intracellular deliverable. We did compare them against controls. So we looked at the size of the nanoparticles versus lipomessenger, which is pretty much lipofectamine, polyjet. And then our size is comparable with Polyjet, which is a polymer-based commercial transfection agent. And our nanogels are actually much better in terms of zeta potential as well compared to these commercial agents. But when we looked at them under the TEM, we can see that the lipomessenger was really aggregatable, messy. So was the Polyjet. But our disulfide nanogels were much more sort of defined. And I may be running out of time, but I will hurry up. So I think it's just clicking, all this clicking. So here we can see that another sort of really interesting finding we found is that our nanogels can transfect cells across different cell lines, but they have this sort of property of having a longer response. And so this green line is basically the commercial one, so the Polyjet mRNA transfection agent. We can see a much higher response, but then it wanes away a lot quicker, which could be related to toxicity. But in a lot of the sort of cases that we investigated, we see the same pattern that across different cell lines, as you can see, all of these lines that are shown here, these are our nanogels delivering GFP mRNA. So it may be that there is some layer by layer effect where the surface is being degraded and the nanogel is coming out over time. And we also measure the cellular toxicity, and we have kind of a sweet spot to work with when it comes to the optimal range. So we were very happy with the concentration, with the transfection data. We can see here cells that have taken up the nanogels and are expressing a yellow protein. And here we can see the normalized data where, compared to lipofectamine, Polyjet, the disulfide-based nanogels are 3.5-fold more effective in transfecting cells compared to even the non-disulfide-based ones. So we've actually looked at this with the Mathiel group in Geneva. We've blocked the sort of extrafacial surface groups on cells, and we've shown that there is an active mechanism involved in the disulfide-based sort of uptake of our nanogels, which is really interesting. And so here you can see on the left-hand side, again, the top panel is the non-disulfide-based image. The bottom is the green nanogels that are based on disulfides. On the right-hand side, you can see tumor spheroids. So these tumor 3D spheroids have been taken up on nanogels, the top two panels. Yeah, and over time, we can see that the disulfide-based ones are working really well. Again, if I just skip to the next slide. So we collaborated, we moved on to functional mRNA. We collaborated with Dave Carling at Imperial and MRC, who works with mitochondria and metabolism. And he had a really nice target for us. This is PGC1 alpha. So if we deliver PGC1 alpha effectively to prostate cancer cells, this allows the mitochondrial biogenesis to go up, fatty oxidation to go up and ATP production to go up. So in a way, we can oppose the glycolytic, but allow this anabolic phenotype that supports this rapid cancer cell proliferation. So by overexpressing this PGC1 alpha, we can suppress tumor progression in prostate cancer models. And that's exactly what Rupali's work showed. So here we can see just uptake of our nanogels in cells, but on the top right-hand side, you can see the very red sort of expressing cells, and that's the cells with the nanogels. So the nanogels are able to express the biomarker, the protein rather, which is the PGC1 alpha in this case. So we were happy with the preliminary results there, but we wanted to allow for targeting. So we looked at the literature, we found a really nice targeting peptide, which we then conjugated using malamide chemistry to the surface of the nanogels. So this is against the prostate cancer, which is the focus actually of this project. So the prostate cancer tumor cells are overexpressing PSMA, and this peptide binds to them. So we then took this further. So Rupali was able to show on the left panel, these are the non-targeted disulfide-based PSMA nanogels, but on the right-hand side, she was able to show that the targeted ones, you get a lot more uptake over time. Perhaps coming to the end of my talk, this is really kind of like a very nice result for us. So we were able to grow prostate cancer cell-based tumors subcutaneously in mice. And then on the left-hand side in this image, you can see the untreated tumors that have been taken out of the mice. And then in the middle, you can see a non-functional mRNA that's been delivered. So it hasn't done a lot compared to the other panels. So and here you can see untargeted PGC1 alpha mRNA nanogels. And then the last sort of line, you can see the targeted PGC1 alpha mRNA nanogels, which means that the system is working. And here you can see the quantification of the tumor volume as shown in blue. So over time, we can shrink the tumor volume, but we've got to keep up the therapy because around 30-something days, the tumor can start growing if we don't keep up the therapy. So what's next in the world of nanogels and what's next for us? So we are now taking this type of technology and platform to make it high throughput. So we're working with machine learning experts and AI experts in our building to basically create nanogel libraries. So we can synthesize nanogel libraries, purify them, look at the transfection efficiency, and basically close the loop in terms of the AI and ML by feeding our data into a cloud computing and using, for example, Bayesian optimization algorithms to actually come up with a nanogel SAR, so a structure activity relationship. And I hope to be able to speak to that soon. So yeah, in the end, I hope I've convinced you that these nanogels, which you can really think about in terms of molecular Lego, they come together in different styles and different pieces, can actually really address a lot of the LNP shortfalls. And we believe, you can use bio-inspired approaches. They are covalent and stimuli responsive. They have existing MP synthesis and scale up and manufacturing. You just need water. You can really tune the size and the porosity of these things. There's a lot of space, chemical space to explore. So in your labs or anywhere where there are sort of cleavable moieties or chemistries to explore, they can very easily come into play if they are water soluble, for example. And yeah, it's a new innovative platform in our hands. So I'd like to thank my lab and these specific students whose work has been presented here, and also my collaborators and the funding agencies. And all of you, thank you very much for listening. And I'd be happy to take any questions.